Okay, welcome to the session 2, which is about the microbes, food and human health, and we will start with enhancing the nutritional quality of food by fermentation by Minna. So please.
Good morning from my side also. Maybe it's still morning before noon. So yeah, like Anne said, I'm going to talk about fermentation and food fermentation and how we can use it to enhance the quality of food. So the work was done in work package 4, and here we had the main aim was to exploit microbiomes for treating both raw materials and the side streams to add the value of these ones. So to reach this aim, 7 of the SIMBA partners were included in this work package and working on it. And I start with the value addition of side streams. So we had the idea to create new or improved methods to utilize also side streams. We focused on tomato, potato, and maize. In fact, in this presentation, I focus only one of those ones, so potato is the subject today. And which kind of side stream? We picked material from the potato starch industry. It was cellular fluid of potato, and we had the idea to cultivate microalgae that could use the nutrients present on this side stream. And the work was done in Niva, Norway, and they screened several strains, the capability to grow in this kind of medium. And after the first screening, 2 strains were selected for further studies. And after characterization, the protocol was upscaled, and as a result, it was found out that the growth rates and the biomass produced were quite similar when compared to this cell fluid and the standard growth medium for microalgae. And also the chemical profiles were studied, for example, amino acids, fatty acids, and carotenoids. They were shown to be quite in the same level in both of these media. So it kind of indicates that this has quite good potential to be used in this kind of approach. And as we know, there are several different kind of exploitation potential for microalgae in different kind of industrial sectors like cosmetics, food, feed, agriculture products. So there is potential for this kind of approach. There is still next steps that should be kind of go through. The cultivation on the commercial scale was not tested. And of course, there is also the cost of nutrients is the limiting factor in this, or the cost of the process itself. But for example, this kind of inexpensive side stream could be one solution to reduce the costs for cultivating the strains. Of course, there is also some regulations and which kind of strains can be used for food and feed applications. Then I move on to the food approaches, how microbes can be used for producing nutritionally enhanced food. And for this one, I kind of introduced you to 2 different approaches. And first one is the fermentation of pulses. As we know, pulses have high nutritive value, and they are recommended as healthy, as part of healthy diet. But what is bad is that they contain anti-nutrients, which kind of decrease the bioavailability of some of the nutrients present in these raw materials. So what we did was that we aimed to optimize microbial consortia to be used in the processing of pulses. And for this study, we selected faba bean, pea, and lentil. And okay, so first we kind of selected the strains for the microbial consortia. We had single strain experiments, and based on those ones, we selected strains for the consortia, and they were consisting of lactic acid bacteria, and 2 of them were also containing propionic acid bacteria. So we were studying totally four different consortia, and we developed processing protocols for those ones, and then we studied the effects of the fermentation on the nutritional content of the final product. And if I start from these anti-nutrients, we aim to decrease the level of those ones. First, we have here galacto-oligosaccharides, which are known to be those compounds causing the stomach problems. And using these microbial mixtures, we could reduce the level of those ones significantly. Then we focus also on lysine-conlysine content, and these are the compounds for certain group of people. They are kind of dangerous compounds because they lack the enzyme which can degrade the degradation products of these compounds. And also in this case, we could reduce the level almost to 0. Then we also studied how the fermentation affects to the tannin content, which also are compounds that reduce the bioavailability of proteins or minerals, and we got a slight decrease also on the level of tannins. And secondly, we focused on the bioactive compounds, and we aim to produce vitamin B12, which is not usually present in the plant-based material. So 2 of the mixtures, microbial mixtures, contained also propionic acid bacteria, which are known to produce active form of the vitamin B12. And as a result of using those 2 mixtures, we could produce active form of vitamin B12. And if we think about the level, I could say that 1 cup of the product would be enough for the recommended level of the intake of vitamin B12 in daily dose. So it was quite nice level that was produced. Then we also found out that the level of gamma aminobutyric acid was increased using this consortia. And then we focused also on the protein fraction and peptide production, and we had the methods to study the protein peptide mixture, and based on the study, we found positive effect on protein digestibility. We also targeted on the bioactive peptides. There is a methodology that can be used in silico, but also in vitro methods, and we found out new bioactive peptides in the mixtures. And also along the way in University of Parma, they have updated the peptide database for plant-based materials, and here's some figures and which kind of bioactivities those peptides may have. And then we also made some smaller scale upscaling to produce the information for the bigger scale processing of the materials, and that was done in work package 6. Then we wanted to also see how these fermented products work in real products. We made prototypes and tested the taste and texture, and so we have kind of a yogurt type product, fresh cheese like product, and fresh porridge type product that were tasted in our tasting panel. And also the products were studied in vitro digestion studies in NMBU Norway. And what we found out was that this plant-based food protects the microbes present in these products, and they are still metabolically active after the digestion. And they can still further work and reduce the anti-nutrients and help digestion. Also, these metaproteomic methods were applied to the samples collected from the in vitro digestion in different phases of this one. And then another kind of approach made in fermentation experts. There was a bit different type of microbial consortia consisting of lactic acid bacteria, and the materials that were used were rapeseed, seaweed, pulses, cereals, and also in this case decreased level of anti-nutrients were detected. Galactolisaccharides and glucosinolate levels were decreased. There was enhanced level of bioactive compounds like omega fatty acids, benzoic acid derivatives, and also there was effect on amino acid profile. It was also tested the large-scale protocol and to produce these fermented ingredients, and so that there is material enough to be tested by customers. And in fact, there was shown that there is positive effect on the shelf life when these ingredients were added to the products. And what we see, there is exploitation potential of this. We have these consortia which can be used for to produce higher nutritional quality food. We have procedures, we have different kind of methods that are available for stakeholders, and we have these models that can be used kind of basis for the further food development, because they are really prototypes that we had in this case. What we saw, there is impact on the scientific part, but also we can see some longer term impact on the industry. There is new knowledge towards the market needs for healthy, sustainable products. This might have a part to impact to the variety, increased variety of these plant-based products in the market, and the way to make the product in a sustainable way. And kind of we identified similar types of impacts for the society. Customers have more possibilities to select products, and they are available for all the customers, and they are made in an environmentally way or friendly way. So I think I can finish with this one. Thank you for all of you.
We have time for one question here.
First, I would like to say fantastic work. Second, what we tried to do was to actually not use in pulses and rapeseed, but microbial upgrade of the side stream from pulses after protein extraction and after rapeseed after oil extraction.
Yeah.
And if you're interested, we can share results. My question is the microbiome you are using here for the last part you presented, are they regulatory approved for microbiome, for food use?
I can say from the first part that all the microbes we selected for the pulse fermentations, they are in the QPS list. But I think Ninfa... Okay, so also the other part for the rapeseed, seaweed
the
part.
microbiome is accepted for fermentation. Is that what you have regulatorily approved?
Yes, and it's not rapeseed, it's rapeseed meal. Sorry, we just say rapeseed. So it is the meal of the extraction of the oil.
Well, there's a heck of difference, though.
Yeah, it's true.
It's rapeseed meal.
But fantastic. But are you all, I mean, you are sure it's on the list? I have not found microbiomes on the Danish food regulatory to be approved for food, but maybe they are.
We have found that out actually. Yeah. So I have somewhere a document.
But then put it in your presentation, it's fantastic.
Thank you very much. So, would you happen to know if there's any overlap in the fermentation with your consortia, and whether or not the indigenous human gut microbiota are able to do some extent of this conversion? For instance, the soybean oligosaccharides, they are typically fermented by the gut microbiota, and you indicated they are also fermented by yours.
Yeah.
So, but have you looked at this?
No, I haven't looked, and I think Davide, have you something to add to that?
Thank you.
I think we haven't had this kind of study. Thank you.
Okay, thank you once more, and I think that we have now to proceed.
Thank you for all of you.